The process of converting DNA into protein is known as protein synthesis or translation. It involves several steps, including transcription and translation. Here's a simplified explanation of the process. Transcription. The first step is the synthesis of a messenger RNA, mRNA, molecule from a DNA template. This process occurs in the nucleus of eukaryotic cells or the cytoplasm of prokaryotic cells. The enzyme RNA polymerase binds to a specific region on the DNA called the promoter and begins to unwind the DNA helix. The RNA polymerase then uses one of the DNA strands as a template to synthesize a complementary mRNA molecule. The mRNA molecule is synthesized in the 5' to 3' direction, with complementary base pairs, A with U, T with A, G with C, and C with G. mRNA processing, eukaryotes only. In eukaryotic cells, the newly synthesized mRNA molecule undergoes additional processing steps before it is ready to be translated. This includes the removal of non-coding regions called introns and the splicing together of the remaining coding regions called exons. The modified mRNA, known as mature mRNA, is then transported out of the nucleus and into the cytoplasm. Translation. The mature mRNA molecule interacts with ribosomes, which are the cellular structures responsible for protein synthesis. Translation occurs in the cytoplasm and involves the decoding of the mRNA sequence to synthesize a protein. The mRNA is read in groups of 3 nucleotides called codons. Each codon corresponds to a specific amino acid or a stop signal. Transfer RNA, tRNA, molecules. tRNA molecules are small RNA molecules that carry amino acids to the ribosome during translation. Each tRNA molecule has a specific anticodon sequence that is complementary to a codon on the mRNA. The tRNA molecule binds to the corresponding codon on the mRNA, bringing the specific amino acid to the growing polypeptide chain. Protein synthesis. As the ribosome moves along the mRNA molecule, it reads the codons and recruits the appropriate tRNA molecules. The ribosome catalyzes the formation of peptide bonds between the amino acids, linking them together to form a polypeptide chain. This process continues until a stop codon is reached, signaling the end of protein synthesis. Protein folding and modifications. After translation, the newly synthesized polypeptide chain undergoes further modifications and folding to become a functional protein. This may include the addition of sugar molecules, glycosylation, the removal of certain amino acids, cleavage, or the formation of disulfide bonds between cysteine residues. The folded protein then carries out its specific function in the cell. It's important to note that this is a simplified overview of the process, and there are additional factors and regulatory mechanisms involved in protein synthesis in living organisms.